says, I call this the molecule movement. It states as compare the mechanisms that transport materials across the plasma membrane. That's just another name for the cell membrane. Passive transport, diffusion, osmosis, facilitated diffusion, and active transport, pumps, endocytosis, and exocytosis. And so this is a nice drawing that will help us keep track of all of these different ways for molecules to get across the membrane. Of course, maybe I'll use our highlighter here. There's our cell membrane, right? So that blue structure in the middle, you guys recognize, that's a cell membrane. And let's say these green balls represent things that are dissolved in water. So let's say they're sugar, for example, okay? Which side is higher in concentration? The outside or the inside? Let's see here. There's outside and there's inside. Where do we see more of the dots? On the outside of the cell in this drawing or the inside of the cell? Owen? The outside. And so what we could say is that outside has a higher concentration than inside. Remember, we think of concentration as how crowded an area is with molecules. So it's pretty crowded, right? There's a lot of green balls hanging out there outside of the cell. And inside the cell, there's a lower concentration. There's a lower concentration inside the cell. Now, they could have drawn it the opposite way, but that would have been fine. But this is how they drew it. So we're going to be referencing high and low. Good, Owen? I like that. Yeah, it's kind of like a castle. All the peasants, they're not allowed inside the castle, right? And, you know, they're the plebes. They're the people that get slaughtered outside the castle walls. You're right. They're the ones out there. It's crowded out there, right? It's only nobility that gets to be inside the castle, right? All right. So let's take a look. We're talking about how do these molecules get across the membrane, and there's a couple of different ways. First of all, let's think of this as passive or active, right? Passive, remember, maybe I'll just jot it over here. Passive is by diffusion. The definition for diffusion is basically when molecules go from where it's crowded to where it's less crowded. So I used H for high and L for low. So anytime we see molecules going from high to low, that's diffusion and that's passive. Remember the analogy we used was you're like riding a bike. If you ride a bike from the high part of the hill to the low part of the hill, downhill, do you have to pedal? No, you don't have to, right? It's very passive. You just got to balance the bike and hold on to the handlebars and hope for the best, right? So high to low is passive in diffusion. And so let's look at our diagram. We said there's a couple of different ways that diffusion can happen. Let's draw an arrow to this one. So we're drawing an arrow to an arrow. One way is that molecules can just squeeze between the phospholipid bilayer. They can only do that if they're really small. So imagine a picket fence. Say our phospholipid bilayer is a picket fence, and there's a chihuahua. Is that picket fence going to stop the chihuahua? No, Chihuahua's going to come right out, and they would probably kill you, but they're small. So it's good that they're small, but I've never met a Chihuahua that didn't want to kill me. So they're just vicious animals, right? So simple diffusion, let's go ahead and circle that. That's called simple diffusion because the Chihuahua doesn't even notice the phospholipid bilayer. It doesn't notice the fence. It just simply diffuses from high to low. That's actually not true. Chihuahuas are great dogs, but I think they're kind of like a one-person dog, right? They just don't like any other people around. They're human. Okay.
Can I say something about Chihuahuas?
Yeah.
They made them in Beanie Boo form.
What'd you say?
And they even made Chihuahuas Beanie Boos.
Beanie Boos. Oh.
But added funky colors and a nice furry collar.
No respect for Chihuahuas. No respect, right?
But they even made a special Chihuahua with a unicorn horn. It's a Chihuahua horn. And her name is Yes.
Okay, great. Let's go on to this next one. Do you see what's happening here? Is this diffusion, the one I just pointed at? I'll circle it here, right? That's diffusion too, right? It's going high to low, so it's still passive, but this time it's having to go through a gate protein. And so when that happens, we call that kind of movement facilitated diffusion. Remember, to facilitate is to help. So this is diffusion with the help of a gate, right? Maybe this is like a golden retriever, right? The golden retriever, the picket fence is going to keep the golden retriever in, right? But if you let them out the gate, then they can get out of the yard, right? And so that gate protein is like the golden retriever, allowing molecules across. Again, it's still high to low, right? Okay, let's look at one more. Remember, we said passive is high to low, active, on the other hand. Any guesses? Not high to low, but low
to high.
Low to high, right. And the analogy I like to use for this is shepherd dogs. Have you ever seen a collie or an Australian shepherd, right? These dogs concentrate sheep into small little pens, right? But it takes a lot of energy for them to do that, right? They really have to work hard to get the sheep to obey them and to get crowded into their little pens. They like to spread out, but they are forced into their little pens for their safety. Let's go ahead and draw an arrow over to here. That's what we see pictured here, right? It's going from inside the castle to outside the castle, right? So somebody just got kicked out to be with the peasants, right? They went from the low side to the high side in terms of crowdedness, not in terms of affluence and wealth, right? They just got impoverished there. But I imagine that sort of thing happened, right? Not in the club anymore. And that requires energy in the form of ATP. So let's circle active transport. We call that protein a pump protein. So that gate right there, it's not just a gate, right? It requires some energy. It's not just a door. It's a door that pushes the solute out. It's kind of like our heart. Our heart is a pump, right? It pushes blood to all parts of our body right now. It's pumping or pushing the solute out of the cell. Maybe that's why it's low inside and high on the outside. Any questions here? So we talked about 3 different ways that molecules can get across the cell membrane passively. There are 2 ways: simply diffusing like a chihuahua, or using a carrier protein and doing facilitated diffusion like a golden retriever. And then we also talked about active transport like the collie or the shepherd dog chasing the sheep and concentrating them into the pen. One that we didn't see in this picture that I want to make sure we talk about too is osmosis. Osmosis is diffusion, but it's when water moves from high water to low water. Okay, and so remember, O for osmosis is O for what? You guys remember this? Opposite. O for osmosis is also O for opposite, because remember, the direction of osmosis is opposite of diffusion. So if those molecules were diffusing in our picture, they're diffusing from the high side to the low side, looks like in through our cells. Which way is osmosis going to want to go? It's going to go the opposite way. So it's going to want to go out of the cell. So let's draw an arrow and we'll label it osmosis. Osmosis is going to want to go opposite the direction of diffusion. Or another way to think of it is the same direction as active transport, but that's a little confusing. So just remember O for opposite. Remember, when water moves into or out of the cell, that changes the shape of the cell, right? If all the water leaves, what's going to happen to the shape of the cell? It's going to get big.
Kramer,
Yeah.
your iPad's low.
Oh, no. Should I stop?
That's why I like to have my Chromebook on the charger at all times when I'm in the Google Meet.
Nice. What should I do?
Plug it in.
Okay.
if
I
you
don't know.
bought your iPad charger.
Yeah.
But have you ever heard of a 10-foot charger? For
Here
the
we are on the
iPad,
second page here, guys. Sorry, did I get too fast here?
Okay, Jenna, let him go. Let him teach. Thank you.
Oh, the other one. This one? Oh, no, this one. Or this one. Yeah, okay, got it. So we just highlighted osmosis as O for opposite. Okay. And then this next page has some pictures. Remember, water is what determines the shape of the cell because it's the most abundant molecule. So if water leaves, it shrivels the cell. So hypertonic, we said shrivels, the cell shrinks. Isotonic, the cell is going to move in and out and it's going to stay the same. Hypotonic, that means that water is going to move into, where does it say that? Cell expands and may finally, well, expands. So we'll enter and expands. I don't think you need to worry about the plant, so we can just cross that out, but it basically does the same thing, except the plant has a cell wall, which is rigid and doesn't flex. Okay? That was kind of a lot. Oh my, we're not done. I just remembered endo and exocytosis. Look at that. Endo and exocytosis. Remember we said, what if there's an elephant in our yard? Is the elephant going to fit through the gate? No. What are we going to do? Never mind how it got there. But if it's going to get out of the yard, we're going to have to tear down the fence, right? Is that going to take energy? Always, right? It always takes energy if you have to tear down the fence. And so remember you said sometimes that's what we have to do. The cell membrane has to tear a hole in itself and allow these molecules in or out. If it's in, we call that endocytosis. Let's underline endo. And let's
remember, endo, enter, exo, exit.
Right, that's a great thing to say. Say it again, Jenna. Can you say it again, Jenna?
Endo, enter.
Good.
So, exit.
You got it. So just remember, EN for enter, EX for exit. That's really handy. That's a good way to remember that. And so this is for really big things that need to get in and out of the cell. Okay, I think we're finally done with that anchor. Yeah, and we're done with that. So let's go ahead and submit this KB